In the vast universe of medicine, discoveries often take us by surprise, revealing hidden connections and unexpected applications. Mefloquine, a drug initially developed to combat malaria, has emerged as a potential weapon in the fight against cancer. This unexpected journey from fighting parasites to targeting cancer cells highlights the interconnectedness of scientific exploration. Mefloquine's story began in the 1970s as a response to the growing threat of drug-resistant malaria. Developed by the Walter Reed Army Institute of Research, it quickly became an important part of our arsenal against this deadly disease. However, the story of Mefloquine was far from over. Researchers began to uncover hints of its potential beyond malaria. These hints pointed towards an unexpected target: cancer cells. This revelation sparked a wave of excitement and curiosity within the scientific community. The journey from malaria drug to potential cancer treatment is a testament to the power of scientific curiosity. It reminds us that the most groundbreaking discoveries often lie hidden in unexpected places, waiting to be unveiled.
Dr. Mahadeo Sukai, a scientist at the BC Cancer Research Centre, made a pivotal discovery that propelled Mefloquine into the spotlight of cancer research. My research focused on acute myeloid leukemia, a particularly aggressive type of blood cancer. My team and I discovered that Mefloquine exhibited a remarkable ability to target and eliminate AML stem cells. These stem cells are the root of AML's resilience and are notoriously difficult to eradicate with conventional therapies.
This breakthrough was significant for several reasons. First, it identified a potential new weapon against AML, a disease with limited treatment options. Second, it highlighted Mefloquine's unique ability to target cancer stem cells, a crucial factor in preventing cancer recurrence. Dr. Sukai's discovery ignited a flurry of research into Mefloquine's anti-cancer properties, paving the way for a deeper understanding of its mechanisms and potential applications. To understand how Mefloquine combats cancer, we need to delve into the microscopic world of cells. Within these tiny universes, Mefloquine disrupts a critical process essential for the survival of cancer cells, particularly cancer stem cells. The key lies in a cellular structure known as the lysosome. Lysosomes act as the cell's recycling center, breaking down waste products and cellular debris. They are essential for maintaining cellular health and balance. Mefloquine interferes with the delicate balance within lysosomes, causing them to become overloaded and dysfunctional. This disruption triggers a cascade of events leading to cell death, particularly in cancer stem cells that rely heavily on efficient lysosome function. This unique mechanism of action distinguishes Mefloquine from traditional chemotherapy drugs, which often target rapidly dividing cells but spare cancer stem cells. This difference suggests that Mefloquine could potentially prevent cancer recurrence by eliminating the source of tumor regeneration. Lysosomes, often overlooked, play a critical role in the survival of cancer stem cells. These resilient cells rely on efficient lysosome function to withstand the harsh conditions within the tumor microenvironment. Think of lysosomes as the garbage disposal system of a bustling city. In a healthy city, waste is efficiently removed, maintaining order and cleanliness. However, in a city overwhelmed with garbage, the system breaks down, leading to chaos and dysfunction. Similarly, cancer stem cells produce a large amount of cellular waste as they rapidly divide and adapt to their surroundings. Lysosomes are tasked with removing this waste, allowing the cancer stem cells to thrive. Mefloquine's ability to disrupt lysosome function effectively throws a wrench into this delicate system. By overwhelming lysosomes, Mefloquine tips the balance, leading to the demise of cancer stem cells. Section 5: Beyond AML, Mefloquine's promise in other cancers. While initially investigated for its effects on AML, Mefloquine's potential extends far beyond this single type of cancer. Researchers have explored its effectiveness against a variety of cancers, uncovering promising results that fuel further exploration. Laboratory studies have demonstrated Mefloquine's ability to inhibit the growth and spread of various cancer cells, including those responsible for gastric, prostate, colorectal, cervical, and liver cancers. These findings suggest that Mefloquine's lysosome-disrupting mechanism may hold promise across a spectrum of cancer types. Furthermore, Mefloquine has shown potential in targeting chronic lymphocytic leukemia, or CLL cells. This type of blood cancer is characterized by the accumulation of abnormal lymphocytes in the blood and bone marrow. Mefloquine's ability to target these cells highlights its versatility in addressing different hematological malignancies. The breadth of Mefloquine's potential applications underscores the need for continued research and clinical trials to fully understand its capabilities and limitations in the fight against cancer. Section 6. Gastric cancer disrupting growth and spread. Gastric cancer, a disease that affects the lining of the stomach, poses a significant health threat worldwide. Mefloquine has emerged as a potential therapeutic agent against this challenging disease, demonstrating the ability to disrupt the growth and spread of gastric cancer cells.
Laboratory studies have revealed that mefloquine can inhibit the proliferation, or rapid multiplication, of gastric cancer cells. This inhibition directly targets one of the hallmarks of cancer: uncontrolled cell growth. Furthermore, mefloquine has been shown to reduce the ability of gastric cancer cells to migrate and invade surrounding tissues. This finding is particularly important because the spread of cancer, known as metastasis, is a major contributor to the disease's lethality.
While these findings are promising, it's essential to acknowledge that laboratory studies represent an early stage of research. Clinical trials are necessary to confirm these effects in humans and determine the optimal dosages and treatment protocols for gastric cancer patients. Section 7. Prostate cancer inhibiting tumor development. Prostate cancer, one of the most common cancers in men, is characterized by the uncontrolled growth of cells in the prostate gland. Mefloquine has shown potential in preclinical studies for inhibiting the development and progression of prostate cancer.
Research has revealed that mefloquine can reduce the growth of prostate cancer cells in laboratory settings. This effect is thought to be mediated by mefloquine's ability to induce cell death and inhibit cell cycle progression in these cancer cells. Furthermore, mefloquine has demonstrated the ability to suppress the growth of prostate cancer tumors in animal models. These findings provide further support for its potential as a therapeutic agent against prostate cancer.
While these preclinical studies are encouraging, it's important to note that further research, including clinical trials, is needed to determine mefloquine's efficacy and safety in humans with prostate cancer. Section 8. Colorectal, cervical, liver, and CLL promising results. Mefloquine's potential extends to other cancer types as well, showcasing its versatility as a potential anti-cancer agent. Promising results have emerged from laboratory and preclinical studies investigating its effects on colorectal, cervical, and liver cancers, as well as chronic lymphocytic leukemia, or CLL. In colorectal cancer, mefloquine has exhibited the ability to inhibit cell growth and induce cell death in laboratory models. These findings suggest that it may have a role to play in combating this type of cancer, which affects the colon and rectum. Mefloquine has also demonstrated potential in preclinical studies involving cervical cancer. Research suggests that it can inhibit the growth and spread of cervical cancer cells, highlighting its potential as a therapeutic option for this gynecological malignancy. Furthermore, mefloquine has shown promise in combating liver cancer, a complex and often aggressive disease. Laboratory studies have revealed its ability to reduce the viability and proliferation of liver cancer cells. In the realm of hematological malignancies, mefloquine has shown encouraging results in targeting chronic lymphocytic leukemia cells. This type of blood cancer is characterized by the accumulation of abnormal lymphocytes, and mefloquine's ability to target these cells highlights its potential in addressing blood cancers beyond AML. While these findings are promising, it's important to remember that they stem from early-stage research. Further investigation, including clinical trials, is crucial to confirm these effects in humans and establish the safety and efficacy of mefloquine for treating these cancer types. Section 9. A powerful combination, mefloquine and artemisinin. The fight against cancer often requires a multi-pronged approach, and combining different therapies can enhance their effectiveness. In the case of mefloquine, researchers have explored its synergy with other anti-cancer agents, particularly artemisinin. Artemisinin, a compound derived from the Artemisia annua plant, has gained recognition for its anti-malarial properties. Interestingly, like mefloquine, artemisinin has also shown potential in combating cancer cells.
Studies have revealed that combining mefloquine and artemisinin can create a synergistic effect, enhancing their individual anti-cancer properties. This combination has shown promise in inhibiting the growth and inducing the death of various cancer cells, including leukemia, breast cancer, and colon cancer cells.
The synergistic potential of mefloquine and artemisinin highlights the importance of exploring combination therapies to maximize their impact on cancer cells. Further research is needed to optimize the dosing and scheduling of these drugs for different cancer types. Section 10. The shadow of neurotoxicity, a note of caution. While mefloquine holds promise as a potential anti-cancer agent, it is crucial to acknowledge the potential for adverse effects, particularly neurotoxicity. Mefloquine has been associated with neuropsychiatric side effects, some of which can be severe and long-lasting. Common side effects of mefloquine include dizziness, sleep disturbances, anxiety, and vivid dreams. In some cases, more severe neuropsychiatric reactions have been reported, including psychosis, hallucinations, and seizures. The exact mechanisms underlying mefloquine's neurotoxicity are not fully understood, but it is thought to involve its effects on neurotransmitters in the brain. The risk of experiencing these side effects varies from person to person and may be influenced by factors such as genetics, medical history, and dosage. It is essential for healthcare professionals to carefully consider the potential risks and benefits of mefloquine before prescribing it to patients. Patients should be closely monitored for any signs of neuropsychiatric side effects, and the drug should be discontinued immediately if any concerning symptoms develop. Section 11. Conclusion. Balancing hope and caution. Mefloquine's journey from an anti-malarial drug to a potential cancer treatment is a testament to the serendipitous nature of scientific discovery. Its unique mechanism of action, targeting the lysosomes within cancer stem cells, offers a glimmer of hope for combating cancers that are often resistant to conventional therapies. The promising results from laboratory and preclinical studies, demonstrating its effectiveness against a range of cancer types, fuel optimism for its future role in cancer treatment. However, it is crucial to temper this hope with a healthy dose of caution. Mefloquine's potential for neurotoxicity, particularly its neuropsychiatric side effects, cannot be overlooked. Careful patient selection, meticulous monitoring, and prompt intervention in case of adverse reactions are paramount to ensuring patient safety. The future of mefloquine in cancer treatment hinges on further research to elucidate its mechanisms, optimize its efficacy, and minimize its toxicity. Comprehensive clinical trials are essential to determine its true potential and establish its place alongside existing cancer therapies. As we continue to explore the vast universe of cancer research, mefloquine serves as a reminder that even within familiar tools, hidden potential awaits discovery. By approaching this potential with a balance of hope and caution, we can navigate the delicate path toward developing safer and more effective treatments for this devastating disease.